All right. Welcome, everyone. Welcome to Hector 2021 and this long workshop session on trajectory inference. We'll start the workshop shortly, but first a few housekeeping items. If you have questions for Hector, then you can go ahead and just enter them into the Q&A tab. He might also ask you like how you're doing and stuff like that, and you guys can use the emoticon features for like thumbs up and things. If questions get entered into the Q&A tab, you can upvote the questions so that we can answer the ones that are most interesting to everybody first when we get a chance to talk about those. And if you would like to ask your question live, just raise your hand. There's a little raise hand tab, and we'll bring you onto the stage so you can ask it live. And if for some reason you guys are going to have to leave early, there's going to be a video available of this session a couple hours after the session is done, and the location of the workshop files that you'll be connecting to will be around for a little while for you guys to enjoy the workshop information longer. Questions are going to be answered throughout, so just post them up there immediately, and then when there's an opportune time to answer them, Hector will answer them. And if you have some more questions after the session is over, you can reach out to him through the AirMeet messages feature, and just feel free to connect that way. So without further ado, it's my pleasure to introduce Hector Roux de Bezieux, I believe is how you say that. Sorry if I butchered that. From the University of California, Berkeley, who will be teaching us about trajectory inference across multiple conditions. And please take it away, Hector. Thank you.
Thanks for the introduction, Glenn. I'm going to be posting right now on the chat the link for the workshop, and then the link for the Orchestra app, which will allow you to run the workshop interactively. And let me do a quick demo of how to find the workshop, because there is more than one. My screen should appear in a few seconds. Yes, great. So if you go to the Orchestra, there's a search at the top, and you can just look for trajectory. And please launch the most recent workshop, the link from 2021. So you just click on launch, and I'm not going to click it because I've already have a session opened, but you can do the same thing and then follow the instruction. So I'll give a few minutes for everyone to start doing this. And while we launch, I can just go through the introduction for the workshop. If you have trouble launching it, just ask question on the chat or the Q&A. So before we actually go to the workshop itself, just to give a little bit of background, I'll be presenting today on trajectory inference across conditions. And Kelly Street, who's a former member of my lab in Berkeley and now a postdoc at the Dana-Farber Cancer Institute in Boston, is going to be presenting with me and helping answer all the questions. And we also worked on that project with Koen van den Berge, but he's in Belgium right now, and he fell asleep following the second dose of the vaccine, so he won't be joining us tonight. This project started, I think, a year and a half ago now, maybe 2 years ago, by a GitHub question on our previous work, which was TradeSeq. Someone asked for a new feature, and it launched a full conversation. We actually started helping them on the data set, and then we created a full package and a paper to go with it. And this workshop, just from someone asking a question on GitHub. So I think it's I've been sharing that story a few times, but I think it's a really cool demonstration of how open source software really helps you have new ideas and then develop them on real applied projects. And so I'm going to be presenting two data sets today as example, and we're going to be going through them together. And the first one, I'm going to first talk about the experimental design. There's a paper from 2019 from McFarlane, Figueroa, and others. And they start by doing some, well, they do in vitro experiment with some epithelial cell in the middle. And as they grow, they're going to migrate outside the plate and differentiate into mesenchymal cells. So this is the experimental design. And they repeat this experiment twice, once in a control condition, which is called mock, and then under treatment, which is called TGF-beta, which is a growth accelerator.
Hector?
Yes.
We have a request that you make the slides full screen.
Oh, yes, sorry. Perfect. Okay, so let me just go through this, the slides again. So you can, yeah, actually this one especially was particularly small. So this was the GitHub question. This is the experimental design. You can find the paper here. We also have a link to the paper on the vignette, so you can easily find that paper again. And then this is the full experimental design with the two conditions. And so this is what we're going to be presenting. So before I launch into the workshop itself, I'm just going to do a poll to see if everyone has managed to launch. So if something's going to pop up on your screen, if you've managed to launch, press yes or no, and I have some idea of what's going on. So I imagine the remaining people are still trying to launch as well. So I'll give it a couple of minutes. If you have issues, don't hesitate to ask on the chat. Give it 1 more minute, and then we can. Okay, I'm gonna start. So the first, so you're gonna pop up on an RStudio IDE interface, and the first thing you can do is go to Vignette, and then click on the TGF-beta vignette, which is the first one we're going to explore. If you want to see a compiled version of the vignette, you can go to the website I've posted the link on the chat, and then you can find the vignette here. So articles, first example, TGF-beta datasets. And it should pop up. And so here you will see everything, all the plots will be appearing as we go on. If you want to run the code as I present it, you can also use the app. So let's go through it. So we're going to be going the analysis in 3 steps, which we're going to call differential topology, differential progression, and then differential expression. And the idea is to be going from very large scale and then going to more and more finer analysis at the gene level afterward. So there is quite a lot of packages to load at the beginning. You can just do that. And I've already presented data set. So we have 2 treatments, MOC and TGF-beta, and we're going to be studying that and try to contrast, see what the impact of the treatment has been. The first thing to do in that case is to do some normalization and batch correction. This takes quite a long time, and so I'm not going to run that code, but you've got all the code to actually produce an analysis. You can download the data set from Geo and then do the integration with Seurat. If you also clone the package, which you can do from GitHub, there is also the dataset itself is already going to be, let me just clear everything. The dataset is going to be already in the package, so you can just load it here. And the first thing we're going to do, so this is already going to be after integration. And the first thing we're going to do is do some visualization to see what the dataset looks like. So it can seem a bit slow at the beginning. That's because of something called lazy loading, where you can see the TGF-beta here is called promise. If you're not familiar with that, it just means that at the beginning, it's not really been fully loaded into memory. And so that's why this function that might seem quick is actually a bit slow at the beginning, because it's just loading the data set. So let's take a look at what we have. So this is a UMAP representation of the data set. And we've got the 2 treatment MOC in green, TGF-beta in purple. We can also visualize the developmental pattern. And so we can see in dark green, we've got the cell that were at in the middle of the plate, as I showed at the beginning, and then afterward, the cell that migrated to the outer of the plate. So we can see we have way more cells on the left side at the inner part of the plate, and then many more cells at the end, the outer part. So here we've got a clear trajectory, and so we might want to fit a trajectory to that data set. There is one lineage, there is one path of development, there's no branching or anything. But the question we might ask is, should we fit a common trajectory to the full data set, or should we fit one trajectory per condition? Because the treatment might make a big difference on the development, and maybe there is a different trajectory, there is a different path that's being taken because of the treatment. And so if we try to answer that question with a few tools, and the first thing we have is a visual tool called the imbalance score. And so it's a function in the condiment package here. And we can run it. And so what the imbalance score, sorry, I said condiment score, but it's supposed to be imbalance score. What the imbalance score is doing, it's looking at a region in this UMAP plot. You can specify whatever dimensional reduction method you want. And it's going to look for imbalance in distribution of the cells, of the treatment, sorry, of the conditions, so treatment and control in that case, compared to the full distribution. And so once you've done that, you can actually plot the score, and you're going to see region with very high score are going to be region of high imbalance, and then region with low score are going to mean we have an even distribution compared to the global distribution. So let's take a look. And so here we can see the score is pretty much homogeneous nearly everywhere. We just have a small region here with very high score. And if that happens, it's usually, but not always the case, especially it's usually because we did not perform the integration perfectly. So there might be one region where there is some imbalance, but it might be linked to the batch effect. Especially here because it's not along the developmental path. That could be reassuring that we actually can fit a common trajectory. But all of this is very visual, and it's hard to get a principled approach for that. And so we've also developed a test to actually see if we should fit a common trajectory or not, which we call the topology test. And the topology test is going to fit a trajectory to each condition, and then do some random permutation of the condition level and refit trajectories and see how different that is compared to fitting one common trajectory. And so to do that, first, we can run slingshots on the dataset. So we run the common trajectory, and we can see that the skeleton of the common trajectory here is very simple because it's a one lineage. So we go from the inner cluster to the outer cluster. And then
Hector,
yes,
we have a question about the imbalance score. I don't know how to put it up
on
the page.
Here, I can put that up on this.
Yeah.
Yeah, here, sorry. So it says, when you look at the cell distribution on the dim plot, do you not see a large score using imbalance?
Oh, yeah, that's a very good question. Sorry, I should have been clearer. So the inner outer, well, let me go back one step. So we have two things here. We have the conditions is going to be the treatment label, so MOC or TGF beta. But then we have also this spatial ID, which is inner or outer, and that's going to impact, that's just reflecting the developmental path. And so here in the imbalance score, if you see what we specify as conditions here is going to be the treatment ID, not the spatial ID. And so in this imbalance score here, we don't look at the inner versus outer. But that's actually, we can actually try it out, see what happens if we replace the treatment by the spatial ID. And I hope I don't break everything, but that will give you a very strong imbalance between the two sides. Because those labels are not at all evenly distributed in the reduced dimension space. However, what we did before was contrast the MOC and the TGF beta, and those were well distributed. So let's wait a few seconds for that to compile. Yes, and here we can clearly see we have two big regions of imbalance on the right and on the left, which is where we have only inner cell on the left and then only outer cells here. And then in the middle, as we would expect, we have a region of low imbalance because this is where we have a mixture of both inner and outer cells.
Hector, you have one other question. I'm going to post that off stage. In the region that's dominated by the outer cells, which is the clarification that Karen had for that.
Yeah, I think that was the follow-up to the
perfect
question.
Perfect. There was one other person that was having issues getting to the code. Bogdan, were you able to find it with the link that I provided? I just want to make sure that everybody's able to see it for the workshop.
So there is for everyone, there's a reaction option at the bottom of your screen. So if you can react whatever you want. Okay.
Yeah, that should be.
So
fine.
we have.
They can always get cloned that repository.
Yeah.
Yeah. Perfect. Okay.
So we have this imbalance score. Just as a reminder, I'll show people what the plot looks like before. Just using the compiled version of the vignette. It should be here. Yeah. So here we've got much less imbalance when we look at the treatment ID instead of the spatial ID. And so the more principled approach to knowing if we have to fit a common trajectory is to do this topology test. So the topology test, yeah, as I was saying, is going to be relying on this skeleton trajectory. This might seem a bit confusing now, especially with just one lineage, and we'll have a second example to dive more into this. Because, yeah, the reason we focus on the skeleton just quickly is because you might change the number of lineages if you just run slingshot over and over again, or any trajectory inference method. And so keeping a common skeleton ensures that we have the same number of lineages at every permutation we do. And so we can run the topology test, and I've run it beforehand. It takes, in that case, it takes 4 minutes, so it's not that long, but still, for some people, it might just take longer. And so we can run the topology test. You just specify, so the single cell experiment object, the condition, and then the number of repetitions. So I think here it's 100, which I think is the default setting. And so you get this. It specifies the statistical method we use to compare the distribution, the test statistics, the p-value. So here you can see we failed to reject the null, which means we are going to fit a common trajectory. And you can see this thresh option, which is the threshold. So in that case here, we didn't test against the null. The two distributions are identical. We allow for some distance. So we test that the distance between the two distributions is smaller than 0.01 using the Kolmogorov-Smirnov distance. The reason we test with a threshold is because we want to favor fitting a common trajectory. If you fit a common trajectory, you actually, first, you have many more cells to fit the trajectory. That means your inference is going to be more robust. Secondly, if you fit one common trajectory, it makes downstream analysis easier. It's not impossible to do downstream analysis if you have separate trajectories, but it's just easier if you have a common one. And so for those two reasons, we're going to favor fitting a common trajectory, which is why we test against a null with a threshold. And so we can then look at this trajectory. And so I'm going to plot the curve here on the data set, and the data set is colored by the pseudo time. So you can see you go from pseudo time of 0 on the left, which is where we add the inner cell, to pseudo time of roughly 20 for the outer cells. So for people that have been using the slingshot package for a while, you'll be happy to see, if you're also using ggplot, which is my case, that Kelly has put some new functionalities that allows you to get the curve as a data frame, and then you can use that to plot it with ggplot with this geom path line here. So you can use that code if you've been using slingshot and ggplot to add your curve to your objects. And so here we've got a clear one lineage developmental path as we go from the left to the right. So that's the first step, differential topology. I'm going to pause for a few seconds here. If you have questions, so you can post them on the chat. Okay. So the second step, the second thing we can do once we have this trajectory is to compare how the pseudotime is being distributed between the two conditions. So just because we have a common trajectory does not mean that the cells progress at the same rates between the two conditions. So the first thing we can do is do it visually. So we can extract the pseudotime and we can plot the pseudotime distribution. So first we can color by the spatial ID. And so here we expect some clear distribution differences. As we can see, the cells from the center, the inner cells, are much more heavily distributed at the earlier pseudotime, and then the outer cells are much more heavily distributed at the later pseudotime, which is what we would expect from the biology. Then we can replot those distribution of pseudotime, but color them by the treatment ID this time. And this is where we're going to be able to contrast the two distributions. And so similar to the step one, we first can look at them visually with this plot. And just visually, we can see clear differences between the two distributions. The mock, so the control experiment, we have three modes, kind of an early peak, a mid peak, and a late peak. And in the TGF beta, we don't have this first mode anymore. We seem to have a shift toward the later pseudotime values, which is what you would expect from a treatment that was going to accelerate the growth of the cells. So we can see that visually. We can also test it. So in that case, we're going to be doing a co-organization test, and this is implemented in our progression test. So once again, this is a function in the condiment package. You just specify the single cell experiment object and the treatment. So the vector of conditions, in that case, is the treatment ID. If you run this, you get, so the lineage number, there's just one lineage here, the test statistics, and then the p-value. And we can see there is there's a very small p-value, and so we can reject the null.
Hey, Hector, we have another question in regards to progression test.
Sure.
Kieran is asking, is it just a wrapper for KS mean with some easier options, or is it something more involved with that?
So if you have, in that case where you have only one lineage, it's a wrapper. You can see it as a wrapper. There is also many more, so we can look at the L for the progression test. Bigger for now. So we have a lot of methods to compare the two distribution in the progression test. And if we look at the differences, the default one if you have one lineage, the default one if you have two conditions is the KS, Kolmogorov spin-off test. However, we also have other methods, including the classifier test, which I'll talk about with the second dataset, and which is really useful if you have more than two conditions. There is a permutation test where you just compare the mean of the two distribution by doing permutation of the labels. And then we also implemented the mean maximum discrepancy, which is another non-parametric method using kernel, and that's also useful if you have more than a unidimensional distribution. And that's also the case for the classifier test. So here, because we have only one lineage, the distribution is unidimensional. Because we have only two conditions, we can use the Kolmogorov spin-off test. But in cases where that's not the case, you can use other methods. I hope that answered your question. And the last difference with the ks.test function is that we also have, by default, a small threshold again in that case, because there might just be a small differences in distributions, and those might usually reflect batch effects. And so we test against a threshold again, just to make sure that we find something that's also biologically meaningful. And you can think of that for people that are more familiar with gene differential expression, where instead of testing against the log function of 0, you would test against the log fold change of 1 or 2, for example. This is the same idea.
Hector, we have another question that came up. So KS test tends to exaggerate significance. Have you tried using a negative control to benchmark threshold p-value?
Yeah, so we've done, this is not in the scope of the workshop, but we've got a preprint on bioRxiv where we have some simulation using the DynGen framework. And so we've checked how we do with controlling the p-value if you have no effect. And we have some good control if we have this threshold of 0.01, which is the default. So there is, we have some, we have tested that. I can come back to that later, probably at the end of the workshop if we have some time, on the different statistical tests that we do for the different steps. Kelly, if you can maybe post the link of the bioRxiv preprint on the chat, that might help people. Thank you. Do we have other questions? No. Okay, so we've done the first two steps, differential topology, which is should we fit a common trajectory? Then differential progression, which is do the cell differentiate at the same speed between the conditions. And once we've done that, we can look gene by gene for differential expression. And so we're going to be relying on the TradeSeq package. And so the new version of TradeSeq allows you to run the package with different conditions. The first few steps are going to be quite time consuming. And so they're not going to be run, but all the outputs are saved on the TGF beta object that is coming with the package. So the first step of the analysis would be tuning these parameters, which is the number of knots, with the evaluate k function. So if you're familiar with TradeSeq already, the only thing that will change when you run it in this kind of format where you have multiple conditions is the conditions argument, where you're going to specify the different conditions. And so this is being run here, and then this is going to be the output of the evaluate k function. And so here you can pick the number of knots. It's always a bit tricky to decide the number of knots to pick. Usually we try to pick a value that's kind of at the elbow of this AAC curve. And so here a value of 5 is what we decide on later on. In our experience, if you if you do plus or minus 1 or 2, it has little impact on the downstream analysis. Also, this is kind of not the case here, but we've had many people trying to run those functions with values from 10, 12, 14. In our experience, it's really rare that you need values of k outside, let's say 7, 8, 9 max. And because computation are going to scale with the number of nodes. Those tend to take forever to run if you pick a value above 10. So then we can run the feed-gam function, which is going to run one smoother per gene and per condition. And once again, because that's quite time-consuming, we're not going to run it. And then we can look for gene differential expression. So the first thing we're going to do, and that would be the same if we had no condition, it's just going to look for differential expression along the pseudotime. So here we're going to ignore the two condition, just going to look for genes that vary along the pseudotime. And so we run this association test on the TGF-beta single-cell experiment object. And we're going to test against the log4 change of 2, because we want to find genes that have at least that log4 change. So if we do that, and we look at the genes, we can also do it condition by condition or together. If we do it condition by condition, we can see we have about 2,400 genes that are being differentially expressed in the control, and then about 1,000 that are being differentially expressed in the TGF-beta. We can see how those overlap using an offset plot. So we can see that most of the genes that are being differentially expressed in the TGF-beta condition are also differentially expressed in the mock condition. So this is 8, 4, 9. And then the rest of the genes are mainly genes that are differentially expressed just in the control condition. We can plot a few genes, or we can plot all of them using a heat map. And so here we can see a pattern. I think we've plotted the top. When did we plot? What gene do we pick? Yeah, we plot all the genes that are being differentially expressed in the mock condition. And so this is going to be the heat map. As you can see, most of the genes are either very high and then decreasing, or low and then increasing at the end. And this is something that's been picked up in the original paper as well. So you can see the clear patterns of expression. And then this would be the same thing with the control condition, sorry, the TGF-beta. We can also do gene set enrichment analysis. So here we can run that to find what functions are being found among those genes. So we can run the code using the FGSEA function and package to find those terms. It's going to take a few seconds to run. And we hope to see the epicyclic cell differentiation terms pop up at the top. And while this runs, just a note on the heat map. As you can see, we do some scaling of the expression value. So the way we plot the heat map is first we get a smooth expression value for every single gene using 50 points. And then we scale it gene by gene. This makes it visually much easier to interpret, because otherwise the scale of the colors will be just driven by one or two very highly expressed genes. And so that makes it otherwise very hard to visualize. Yeah. Okay, we can just move forward while this is finishing to run. The second thing we can do, so here, the first thing we've looked at is how do trying to find genes that are differentially expressed along the pseudo-time. The second thing we can do here is to find genes that are being differentially expressed between the two conditions. We're going to look for genes that have one pattern in the mock condition and then another pattern in the control condition. So the first thing we can do is pick up a few genes to look at them from the original paper. And so CDH1 and CRB3, those should have the same dynamics. And so you have here decreasing and then stable. And then for CDH1, different dynamic at the beginning that's been noted in the paper, and then similar dynamic at the end. We can also look at genes that are very strongly different between the two conditions. This FN1 gene and this CDH2. And so FN1, it's very highly expressed. So in the TGF-beta lineage in yellow, but nearly not expressed at all in the mock condition. And then CDH2 had a similar pattern of being only expressed in the TGF-beta condition, but just less difference in terms of absolute values. Let's see if that finished running. Not yet. Okay. I'm going to kill it. It's a bit slow. Just going to show the results in the compiled version. So yeah, if we look at the geoterm that pop up at the top, you can see epithelial cell differentiation, epithelium development. This is what you would expect given the biology at hand. So that was this geoterms were for the genes that are differentially expressed along the time. Now we're back to genes that are looking for genes that are differentially expressed between conditions. So we've got 4 examples, and then we can do the condition test to look for genes to compare the smoothness between the mock and the TGF-beta conditions. And so this is being implemented in the condition test in TradeSeq. And once again, we test against the log4 change of 2. We correct the p-value for multiple testing, and we keep the genes that have a corrected p-value smaller than 0.05. And if we do that, we find about 2,000 genes that are differentially expressed. And we can run it. Again, going to take a few minutes. And then we can plot the most significant gene and the least significant gene. So we would expect a lot of contrast on the most significant gene and not at all on the least significant. The top one, we can clearly see it's really highly expressed in the TGF-beta cells, but not all in the mock cells. And then the least differentially expressed, there's nearly no differences between the two. That's as expected. We can also do a heat map of the gene that are differentially expressed between the conditions. And so here we use again the predict smooth function to look at all the to get a smooth expression of all the genes, and then we get the 2 heat maps. Just another point here on rescaling. So the first time we presented this workshop, we were actually doing rescaling on both conditions separately, but we since changed because we realized if you do that, you're going to have you're going to lose some differences because you're comparing the 2 conditions, and so there should be a common scale for the for one gene between the 2 conditions. And so if you've been using our 2020 vignette, this is one of the big differences for this data set that we've changed at this stage, is to do a common scaling. So if we do that, we've got two clear different patterns between the TGF-beta and the mock condition. One caveat, the reason we have very coherent pattern on the left and they're all on the right is because we actually sorted the genes just using the TGF-beta and not using the mock. That's why it seems much more coherent. But we can still see very big differences between the two conditions, as we would expect. And this is still running the condition test. But then we can do gene set.
Sorry, Hector, I had one quick
Yes.
question. For that heat map, is each
Yes.
row for the TGF-beta and mock, is that the same gene on each row?
Yeah, thank you for the question. Yes, it is the same gene. As you can see here, so the heat map on the TGF-beta, we're going to do, so we're not clustering the columns because we want to keep the pseudotime values ordered, but we do a clustering with this hierarchical clustering of the rows, so one per gene. And then we get here for the mock, we get the order from the heat map of the TGF-beta using this to actually resort the gene in the mock condition in the same way. And so here we'd say you don't cluster the columns, you want to keep the time, but you also don't cluster the rows.
Great, thank you.
Thanks for asking. Still running. Those functions take a bit of time.
We have a couple of questions in chat. Looks like
Yeah.
Kelly's answering them, but I don't know if you want to take a look at them as well. They're not in the QA, they're just in the normal chat.
Yeah.
So let me see. I'm not going to answer the one on gene ontology. It's not the focus of this workshop. However, there is two ones here. So how do you calculate the smooth function? And then how do you identify the precise trends in differentially progressing genes? So the smoothers are being estimated, and let me go back. So if you're following along, that's here about line 250 at the fit-gam step. So we use this fit-gam function in TradeSeq to estimate the smoothers. And under the hood, this function is calling on the MGCV package to fit some spline smoothers to every single gene. So the number of knots is going to be the number of knots for the splines. And that's everything is back to the MGCV package. If you look at the fit-gam function help, it should give you the relevant link to the MGCV package. It should, but it does not give you the links. That's something we need to fix in the help. Okay, so yeah, you can look at the MGCV package to get more detail, but we're fitting an NB, it's a negative binomial general additive model for every single gene using the MGCV package.
It looks like the links are working in the article, just not in the knitted thing. So if you go to your pre-built
thank you.
one, it's there.
Okay, thanks for that. Yeah. So the second question was, the second follow-up was relating on patterns of expression and how do we look for different trends. And here we have not done that. We have just done it. Let me go back. That was linked probably to this heat map where I talked about the different trends of genes that are being like highly expressed and then decreasing, lowly expressed and then increasing. We have a function which is just clustering on gene expression patterns, and under the hood that's relying on the cluster experiment. So that function is cluster expression patterns. And so if you look at that function, you're going to be able to find different patterns of expression that are going or up or down. Yeah, I don't know if we have a quick example here. No, that's taking. This is quite also a huge function to run. But we have something that allows you to find patterns of expression. Do we have any more? No, just that. I think we're good on the questions and the follow-up. So we were at this point, the condition test finished to run for me. And so we can just rerun the rest of the code. Plot the two smooths, the most and the least significant. Then the heat map. Which is just regenerating the same results. And then once again, you can do gene set enrichment analysis. And you can see that the biggest differences come from adhesion, which could make sense because we are migrating on the plate, cell migration, locomotion. All those are terms that pop up as different between the mock and the TGF-beta conditions. And I think that's it for this first example. Do we have question on this so far? Other questions? And so just to do a quick recap of what we've seen, what we've done. So I think they should pop up the, not so much properly, the outline. But what we've done is, so normalization and integration, which we haven't run today, but you can look at the code. Some visualization and exploration of the data. And then first step, this differential topology, should we fit a common trajectory or not, with two tools, the imbalance score and the topology test, which is here. Second step, the differential progression, comparing the distribution of the pseudotimes between the two conditions. So this was this plot, and then the progression test. And then third step, looking for differential expression along pseudotime and then between condition. And so this was those plots, which is along pseudotime, and then those plots for between conditions. Okay, I'm going to pause for a few more seconds for everyone that's following along so your code can finish. If you have error in the code, hopefully not. It should be running exactly the same thing as I am, but you never know. Just ask in the chat.
We have a new question posted. Kieran is asking, why does pseudotime from Slingshot always go a bit over 30 and not further?
Is that specifically in the TGF-beta data?
I think that the follow-up that say I see it in most of my data and here in the TGS samples too.
The units of Slingshot pseudotime are the units of whatever dimensionality reduction you're using. So there shouldn't be any absolute values that keep popping up like that. I don't know. like if you did PCA, it'll be in the units of the PCA space. And if we did UMAP here, whatever arbitrary units UMAP returns are the units along the curve of a slingshot curve.
Yeah, I noticed in the pre-built one, the pseudotime axis is only going up to a bit over 20 and not 30 in this case.
I think, and from like really out of top of my head, the UMAP value seems to be more on the small range of values between, I think here we've got like from -10 to 10 and -5 to 5. And those seem to be always the same values, which is what you would expect. The biggest possible distance you can travel to be are the values of 20, 30s. If you do PCF, for example, where the first few PCs can have much bigger values, you will expect to see a much bigger slow time.
Yeah. Yeah, the other one that I remember using it with was ZinB wave, where the dimensions were scaled. So the pseudotimes would generally be less than 1 in that space.
We can actually look at the second example quickly, see where the value goes. Yeah, you see here they go to up to 200. And this uses ZinB wave plus TSNE. I'll explain later, but just as an example. Okay, so this was the end of the first dataset, the first example. And that gives you a full overview of a full workflow on the dataset. Now I'm going to present a second example. So let me clear that just to free the memory. So this workshop is a bit intensive, so that's good for the computers that run everything under the hood. So the second example we have, and let me go once again, full screen. Oh, the quality is really poor, sorry for that. So from this recent paper that's looking at the reaction of tumors to this new type of drug, which is KRAS inhibitors. And so you have cells in a resting state in a gray cluster, and you can see how when they are presented with the drug, they react along 3 different paths, differentiating into a red, blue, or green state. And so in that paper, they use 3 models of tumor, which is H358, H2122, and SW1573. Kind of have 3 replicates to look at how those tumor models react to the drug. And the paper is very interesting, and I invite you to look at it. But we're going to use it as a different in a different setting. And here we have 3 tumor models, and so we can look at those as 3 different conditions. And so one question we can ask is, do the 3 tumor models react the same way under the drug intervention? So here our conditions are not going to be treatment and control. They're going to be tumor model 1, 2, and 3. And so we're going to have an example with 3 lineages, 3 paths, and then 3 conditions. So it's a demonstration of how the condiment package works in the setting. And so I'm not going to do a full workflow in that case, but just go over a few functionalities there. So once again, we can load the packages. They should all be here. We also provide a function to import the dataset. And we're not going to do differential expression in this second example. So here, I'm not running that, but I'm just keeping the first 100 rows arbitrarily, just so that I can save a smaller object. So if you load the KRAS dataset, it's going to be a much smaller object than the TGF beta, because you don't need to keep all the genes. Joke. So you can see we've got about 10,000 cells, and we've got already one reduced dimension computed, which is TSNE. And so this is from the original paper. We picked the TSNE coordinates because they published. Sorry, this is released, and so we don't have to do the normalization again. We can just pick this reduced dimension coordinate. So let's take a look at what the data set looks like. So first, this is all the cells in TSNE space. So here, back to the question we answered before, you can see the values go from -60 to 60 in the first dimension, and -30 to 60 in the second dimension. And we've got the three conditions, which are the three tumor models. We can also do the same plot, but called by the clusters from the original publication. And so this is what we get. I should actually start with that. If you want to find this vignette, same as before, you just click on Files on the bottom right panel, and then on the cras.rmd vignette, if you're not familiar with RStudio. And let me actually, just going to poll people quickly, make sure everyone is on the cras dataset. Oh, yeah. Please do not try to run this first chunk here. You can see we put eval = false. This is connecting to Geo, trying to download everything. And this hasn't been tested in the orchestral environment. So just keep that chunk of code and just go directly to this one. This has been already processed in the package. So if you can just answer quickly if you manage to have the cras vignette loaded, or if you're not running the code, just using the compiled version, you can find it on the website. Second example, cras dataset. Just going to wait for 2 minutes for everyone to catch up. Okay, so I'm going to go down. So we're going to follow along the first same 2 steps at the TGF beta dataset. First, looking for differential topology. So should we fit common trajectory? So we've got the imbalance score. We can compute it. So here we're going to have 3 conditions, but we can still compute the imbalance score. If it's decided to load.
We're still seeing the false.
Sorry?
We're still seeing the poll. Oh, there it is.
I got it.
Okay. Oh, there is a small delay. Do you see the screen now, Kai?
Yep.
Okay, thank you. So here we've got some imbalance in this region, some clear imbalance. And this is a region which is the end of a lineage. As you can see here, it's the red cluster, which is the end of one of the paths in the original publication. That's already more worrying than the imbalance in the other data sets. And so what we can do here is actually run the topology test. The first thing to do is we fit a common trajectory to all three conditions, all three tumor models. And that's going to be our null hypothesis, which that we have a common trajectory. And then we can plot the skeleton of the trajectory, which is this minimal spanning tree that Slingshot uses. And you can clearly see here, well, it's actually running, starting from the gray cluster, and then one branching to the green path, second branching to the red path, and then long lineage, a long blue lineage. And that's consistent with the original publication. Once again, if you want to get the minimum spanning tree in a format that works with ggplot, you have to specify this as df equal true. And then you can just add the minimum spanning tree to your ggplot object with just those two lines here. It didn't run that check. Let me rerun it again. Here you go. And so what the topology test is going to do is it's going to first fit one trajectory per condition, but keeping the same skeleton, so keeping the same underlying graph. The center of the clusters might change as we look at different conditions, but the tree that's underlying is going to be the same. And so we're not going to, and then you're going to do random permutation of the condition labels, and you're going to be able to compare the distribution of pseudo times when you fit one condition, when you fit one trajectory per condition, and then when you fit one trajectory per random permutation, and see if they are different. And so we can run the topology test here because we have many more cells, and also more lineages and more conditions. It's going to take more time. So we're not going to run it. As you can see, it took 8 minutes to run it before. But this is the result. So because we have 3 conditions, we have to use the classifier test. The classifier test is just training a classifier on a subset of the distribution and then looking at the accuracy in the test set. And so if the null is true that there is no difference between the distributions, you would expect, you can actually know the distribution of the accuracy. So if you have, for example, 2 conditions, you know you cannot do better than 50% at predicting which distribution the data point came from, because if they are identical. In that case, we have 3 distributions, so you can't do better under the null than 1/3. However, in that case, you can see the test statistic, which is the accuracy under the null, is 0.92, so it's a very high value. Accordingly, the p-value is virtually 0, or null of the machine. And so here we reject the null that we fit a common trajectory. We should fit one trajectory per condition. So we can do that. We have a slingshot condition function that will fit trajectory per condition. So you just specify the object and the condition. And then we can plot the minimum spanning tree for all 3 conditions together. And this is what happened. So this is the skeleton of every single trajectory. and then we can plot the lineages and see what it looks like. There's a lot of curves everywhere because we have 3 lineages and 3 conditions. However, you can see here, so here we have one trajectory per condition, but as you can see, the underlying topology is quite similar. And even if you look at the curve, it's still the same. And so we can manually map the lineages one into the other, right? There's like the same lineage 1 here, same lineage 2, same lineage 3. And so even if in our framework here, we don't have a common trajectory, we can still do manual mapping. And so the mapping we're going to do is say all the lineage 1 map to one another, all the lineage 2, all the lineage 3 map to one another. And then we're going to merge the slingshot object and have just one object at the end to do the next step. So because this is the same reduced dimension space, we can still compare. The slow time values are going to be comparable, and so we can compare their distribution. Yeah, 15 minutes. So I'm going to be moving on just to wrap up the next step at a kind of fast pace to leave a few minutes for question at the end. But I'd be happy to talk afterward as well if you have some question, if we don't have time to go through all the questions. So after this differential topology, we can do differential progression. We can look at the distribution of the time between conditions and then along the 3 lineages. So here we have the plot I showed before. Every panel is 1 lineage. Every color is 1 tumor model. And so you can see especially this H2122A lineages, model has much fewer cells at the end of lineage 2. That's kind of the big difference usually we see, but we can definitely see differences between the lineages. We can also do this in a more principled way using the progression test. And here we have an argument which is called lineage equal true, where we're going to do the test at a global level, so test every single, so test for all the lineages together, although we can test one lineage, the lineage 1 by 1. And so if we do the test, all the lineages together, we get a very high test statistics for the accuracy of the classifier test. And then we can do the same for the 3 lineages, 1, 2, 3. The accuracy are kind of comparable. The p-values are very different. That's mostly because we have many more cells in lineage 1 because it's much longer. And so even for quite comparable accuracies, we have a much smaller p-value, which is what you would expect. So here we compared every single lineage individually between the conditions or together. But because we have more than 1 lineage, we can also do a second global test, which we call differential differentiation. The name is not great, and we haven't been able to come up with something better. To give you some idea, what differential differentiation would look like is to say, do the cells prefer going into lineage 1 versus lineage 2 in one condition or in another? So instead of comparing the pseudo-time values here, we're going to be comparing the weights of how each cell is being assigned to each lineage. It's a bit harder to interpret, but we're going to do a few plots. The first one we can do is just look at the mean weight in every single lineage for every single cell and the condition. So here we can see it's kind of consistent with what we've seen before. Lineage 1 has much more weight because there are many more cells in that lineage. And I should have specified the weights are valued between 0 and 1. And the higher weights mean you're more likely to belong to that lineage. And so if we compare the 3 conditions here, we can see especially that the H212A model has much less cells that have high weight in the lineage 2. That's consistent with the distribution before. We can also do, because the weights sum to 1, so technically, even though we have 3 weights, 1 for each lineage, for each cell, because they sum to 1, there's only 2 values we can plot, and then we know the third one is going to be 1 minus the other 2. And so we can plot weight 1 and weight 2, and then we know those for the 3 tumor models, and then we know we can look at those lines. For example, this is where weight 3 equals 0, weight 3 equals 1/3, weight 3 equals 2/3, and then this point here at 0, 0 is just weight 3 equals 1. And we can do an hexagonal plot. So it's kind of a 2D histogram, where the more color we have in the hexagon, the more cells we have at that place. It's hard to do any kind of big takeaway here, but we can clearly see the 3 distributions are very different. And if we do that with the differentiation test, we get this result indeed, that we have either at the global level, or if we do pair by pair, more different. We have differential differentiation between the 3 conditions. And then in normal workflow, we would go on into looking at differential expression of genes between those 3 conditions. But here, we're not going to do it because we don't have the time. But if you want to run it, you can either look at the code of the first vignette and try to replicate it, or we have, for the preprint that we released, all the code is available online at Condiments paper, and so you can find all the code to do also the differential gene expression analysis. Thank you very much for attending today. I'd like to thank Kelly as well for coming, and then Glenn for animating the session. And if you have more questions,
Yeah, that was great. Thank you, Hector. It's really interesting to see all the different types of analysis the package enables us. It looks like there was a question from Kieran. I'm going to put that up onto the stage. He's asking about, we can't do differential gene expression between multiple conditions over pseudotime under TradeSeq, or would you just do condition testing on lineage with the highest weight?
You can do differential gene expression analysis. Definitely, that would be very similar to what you would normally do. Yes.
Showing your clock is making us feel bad for how early it is for you, Hector.
It is a bit early. That's okay. This is the website for the condiment paper. It has less explanation, but if you look at differential expression, after some filtering, we do the evaluate K function similarly, and then you would do the fit gam and fit the smoothest for everything together. So you can actually have, let me see, this will seem similar. So it's a bit more complex because you have one smoother per lineage and then per condition. So you have you have 9 smoothers in total, with the long smoothers being for lineage 1 and then the different groups of colors being for the 3 conditions. But you can definitely do TradeSeq in that case.
It might be worth pointing out that we can only do that because we had the manual mapping between the lineages. If we didn't have like sort of an obvious way to map the lineages onto each other, then we would be in a situation where we couldn't do TradeSeq.
I can actually, we have some toy example as well, just to give some ideas. Of course, it didn't run. Oh yeah, it's coming. Right, just right. Okay, this is ugly, but let's say you've got the blue one. The blue thing is one condition. Just ignore the lines; it should not be coming back. And the orange one is the second condition. So here you can see kind of the knockout has a massive impact on your developmental path. It's kind of very, very strong example just to showcase. But instead of having a branch, you have a third path that's appearing that's only happening in the knockout stage. Not knockout stage. I'm watching too much of this. The knockout condition. And so here you don't have obvious manual mapping, so you will not be able to do a condition test.
All right, that's great. Thank you so much for sharing. Appreciate it. And I think that brings our session to the end, unless there's some last second chat that happens. But thank you very much, Hector, and thank you, Kelly. I really appreciate it.
Thanks everyone for attending.